Hello and welcome back. So today we are going to talk all about matter and energy transformation. So topic 18 is modeling activity, and even though we are doing online lectures, we're going to do our best to still try to make this a hands-on activity. So this is the last topic of our metabolic unit that we're going to cover before we move on to one of my favorite units, which is all about DNA and how we take DNA and copy it to RNA, and then how that RNA is then made into protein. So anywho, I'm getting a little ahead of myself, but before we do all that fun stuff, we've got to finish up our metabolism. So let's go ahead and talk about what our goals are for this activity. So for this activity, we're going to take all of the pieces that we know about aerobic respiration and we're going to synthesize them into one model of how it works. And within that model, we're going to talk about how matter changes, the energy transformations that occur, and then we're going to use our model to then apply it and predict what could happen in different novel scenarios. And then last but not least, we're going to read a Science Daily article that is titled Key Protein and Cellular Respiration Discovered, and we're going to talk about how does what we discussed in class and what we learned in class relate to that article. So on Moodle, I have posted a couple key things for this model. The first thing that is up there is the instruction. So the instructions are up on Moodle, and I encourage you to, before you actually watch this video, or if you are watching it right now, to complete the activity first and then come back to the video and make sure that you are on the right track. So the instructions are posted on Moodle. And the other thing that you will find is this image back here. So this is the scaffold for the activity. And then on the left here, you'll notice that we have all these different shapes and molecules. This is titled Shapes and Molecules on Moodle. So what I would recommend that you do is actually print that out, cut out all of the pieces, and use those to build what's going in and what's coming out at each of the stages of aerobic respiration. So what we're going to do, and what I encourage you to do, is, like I said, cut out all of those shapes and molecules, use those pieces to, in a hands-on way, model what's going on in aerobic respiration, and then that scaffold sheet is our summary of each of the steps. And then we're going to draw and explain the electron transport chain and chemiosmosis. And then last but not least, we're going to discuss the model application and extension questions. And because normally in class, we would do this as a class, we'd have a big class discussion, rather than me going through and doing all of these pieces, a lot of the application and extension questions, we're going to answer and discuss those in the comment section of this video. So I'm going to walk you through how I built my model and how I filled out my scaffold sheet. And so this video will be really short, but again, the goal here is to get you all some hands-on activities to put all these pieces for respiration together. So let's go ahead and start with stage 1. So stage 1 is glycolysis. And in glycolysis, we are starting with glucose. So in those molecules and shapes handouts that we have, each of the gray circles represents a carbon molecule, and so glucose is made up of 6 carbons. So I went ahead and put 6 carbons together, and I summarized glycolysis from the beginning to the end. I kind of skipped all the complicated stuff in the middle, and so at the end of glycolysis, we have 2-pyruvate. And each of those pyruvates are 3 carbon molecules. So we go from 1 6-carbon molecule to 2 3-carbon molecules. While we do that, when we transform that glucose to pyruvate, we are making 2 net ATP, and we're making 2 NADH. So recall when we talked about glycolysis that we said there's an energy investment stage, which requires us to use 2 ATP to make an unstable intermediate, which then breaks in half, and we make 2 G3Ps, which are then converted into pyruvate. So this summarizes our stage 1. And again, you're not seeing it in this video, but I actually played around with all the pieces until I organized it into this fashion. So let's now skip to our scaffold that we have, and I will walk you through how to use this scaffold and kind of how I am using it and how I'm answering all the pieces. So stage 1 is glycolysis, and because I struggle with writing small, I'm actually going to zoom in on the drawing and the explanation for now. So as I draw my drawing, I'm going to kind of take an approach of being somewhere right in the middle. So I'm going to include some details, but exclude some details as well. So I'm going to start with my 6-carbon glucose molecule, and we have 2 stages of glycolysis. So in the first stage of glycolysis, we use 2 ATP to add 2 phosphate groups onto glucose so that we can make it unstable, and those are split into G3P molecules, and those G3P molecules are then converted into pyruvate in stage 2 of glycolysis. And when we do this, we make 2 ATP and 1 NADH, and because we have 2 G3P molecules, we do this twice, and so in our model, that's where the 2 NADH came from and where overall net 2 ATP came from, because remember, up here in this step, we invested 2 ATP. So as an explanation, I'm going to keep this pretty short and sweet, and I'm going to summarize glycolysis as glucose being split into 2 pyruvate. And now that I have my drawing and my explanation done, I'm going to go ahead and zoom back out here, and we're going to fill out the rest of the scaffold. So in this scaffold, we have a couple of different columns. We have ones that allow us to trace our carbon or our matter, and we have a column for ATP, NAD+, and FAD. So in glycolysis, we can actually go ahead and we can cross out FAD because we don't work with that energy management molecule in stage 1. So we're going to focus on the other 3 columns. So for carbon, if we were to go ahead and draw our energy reaction diagram, we start with glucose and we end with pyruvate. Pyruvate has lower free energy available than glucose does. And so when we take glucose and break it down, this is an exergonic reaction, and an exergonic reaction means that delta G is less than 0. We also, overall, so here we're talking about our net energy management molecules. So for our net energy management molecules, we take ADP plus a phosphate group and we make ATP. ATP has a much higher free energy than ADP does. Therefore, this is going to be an endergonic reaction and delta G is greater than 0. And again, for NAD+, we have the same thing. So NAD+ is going to gain an electron, and so it's going to be reduced to NADH. This is an endergonic reaction, and delta G is greater than 0. So again, even here, we can see those dang energy reaction coordinate diagrams that those exergonic reactions are always linked with the endergonic reactions. So taking glucose and oxidizing it to pyruvate, that exergonic reaction is enough to power to make 2 net ATP and to make NADH. And again, there we're making 2. So in our little energy management molecules box that we have, we can actually write that. So we have 2 net ATP and 2 NADH. In the carbon molecules, this is asking us, what does our carbon look like at the end of this step? At the end of this step, we have 2 pyruvate. and those pyruvate have 3 carbons apiece. All right, so this is a really nice summary of stage 1. And again, all we've done here is we have put into words and pictures what we made with our molecules and shapes. So let's go ahead and move on to stage 2. So stage 2 is the formation of acetyl-CoA. So I have in this image actually left stage 1 up. So here is stage 1, our glycolysis, and then here is stage 2. So in stage 2, we are starting with these 2 pyruvate that we have. And for each of those pyruvate, they are converted to acetyl-CoA. Anyone remember what the enzyme is called that does this? Hopefully, you were able to guess that that enzyme is called pyruvate dehydrogenase. And when that enzyme is taking pyruvate and converting it to acetyl-CoA, during that process, we have one of the carbons from pyruvate that leaves as CO2, and we also reduce an NAD+ to NADH. Now, because we have 2 pyruvates, you'll notice that here on this side, we have the same thing happening. So at the end of stage 2, we have these 2 acetyl-CoAs, and we have 2 molecules of CO2 that leave, and then we also have 2 NADH molecules that have been reduced. So we can go back now to our scaffold, and we're going to do the same thing that we did with glycolysis, and we're going to summarize what occurred. So stage 2 again is formation of acetyl-CoA. And I'm going to draw one of the pyruvate molecules. So here is our pyruvate. And pyruvate is converted to acetyl-CoA by pyruvate dehydrogenase. We get CO2 and we reduce a molecule of NAD+ to make NADH. And again, if we were to summarize this, we can summarize this by saying pyruvate was converted to acetyl-CoA using pyruvate dehydrogenase. And now that we've got our drawing, keep in mind we've only drawn one of the pyruvates, so we can actually take this and write ×2 just as a good summary and reminder for future selves that this does happen twice since we have 2 pyruvate. So let's go ahead and zoom back out and draw our reaction coordinate diagrams. So we're going to start with tracing carbon. So although pyruvate is a lower free energy than glucose, it is still a higher energy than acetyl-CoA. So when we draw this, we're going to go ahead and put our pyruvate up here as a product or as a reactant, and our product is our acetyl-CoA. This is an exergonic reaction, which means that delta G is less than 0. So this oxidation of pyruvate to acetyl-CoA is powering our reduction of NAD+ to NADH, which is an endergonic reaction, and delta G is greater than 0. Now you'll notice that I skipped the ATP column. We can go ahead and cross that out. The reason for that is, of course, we don't make any ATP during this stage, and we can cross out our FAD column as well because that energy management molecule does not apply here. So at the end of stage 2, during stage 2, we have made 2 NADH. So if we wanted to, we could actually add a little column here that is our total energy management molecules. So total so far we have 2 ATP and 4 NADH. And then the last thing we're going to fill out is our carbon molecule. So at the very end of stage 2, we have 2 acetyl-CoA, and we made 2 CO2. All right, now we are ready for stage 3. So let's go ahead and I'll show you the picture of the model that I made of stage 3. Come on, there we go. All right. Sometimes when you change pages and you've got pictures, it just takes a while to show up, but there we go. All right, so for stage 3, stage 3 is everyone's favorite cycle, the citric acid cycle. So for the citric acid cycle, just like I did for stages 2 and 1, I actually kept everything up so that we could see how everything interconnects. So here is our stage 1, glycolysis. Here is stage 2. And then everything else on this page is stage 3. And what I have done is, since we have 2 acetyl-CoA molecules, I have shown how each of them goes through the citric acid cycle. So one of the most common questions that I have received is, how do I know how many NADH or FADH2 to count from the citric cycle? Do I count it per glucose? Do I count it per pyruvate? And the answer to that is, it depends on what the question is asking you. If it's asking you per glucose, you're going to want to make sure that you count it per each of your pyruvate. So I like this picture because it shows us that really at the end of stage 2, we have 2 acetyl-CoAs, and each of those acetyl-CoAs has to go into the citric acid cycle. So this acetyl-CoA is going to enter the citric acid cycle. So if we start here on the top, these 2 gray circles that are going into the citric acid cycle, this is our acetyl group. And this is the acetyl group that is left over from glucose. The CoA of HERS leaves the cycle, and this acetyl group is going to join up with oxaloacetate to make citrate, and that citrate gets passed around in a circle. And as that citrate gets passed around in a circle and we knock off carbons off of it, we are going to make different energy management molecules. So following along clockwise, when the first energy management molecule that we make is NADH. So we reduce a molecule of NADH, and when we do that, we knock off a carbon from that acetyl group, so from that citrate, and that carbon leaves a CO2. We continue going around, we reduce another molecule of NAD plus to NADH, knock off another carbon. So at this point, this is the last carbon from that acetyl group. And then as we remake, at this point, our metabolic intermediate to be oxaloacetate, so we can repeat the cycle again, we make another molecule of NADH, a molecule of FADH2, and of ATP. And of HERS, because we have 2 acetyl-CoAs, we do this twice. And so I wanted to make sure that I had 2 citric acid cycles up here to illustrate that this is occurring 2 times. So this is one way that I did it. I was also playing around with some of these pieces another day, and this kind of shows you looking at the stages from left to right, but I also messed around with the pieces and made another model where it's from top to bottom. So again, here we're looking at the same thing. So we're seeing stage 1, we're seeing stage 2, and then stage 3 that we were just chatting about. So just another way to look at it as well. And if you wanted to simplify things, you could also just show this half for the 1 pyruvate and know that it occurs for the second as well. You kind of have complete freedom over how you build this. So let's go back to our scaffold and let's fill out our last row. So again, I'm going to go ahead and zoom in here, and we're going to label this as the citric acid cycle. And let's go ahead and draw a representative drawing of what happens. So I'm going to start with my oxaloacetate, and I'm going to try to draw a nice circle here. There we go. It's about as good as it gets. So we're going to bring in our CoA, or our acetyl-CoA, I should say. Our CoA leaves. That acetyl group enters the cycle. and we make citrate. That citrate is going to go around, and as it goes around, we're going to knock off carbons that came from that acetyl group. They leave as CO2. During this, we will also make NADH. So we make 3 molecules of NADH, a molecule of FADH2, and 1 ATP. And if we were to summarize what happens in the citric acid cycle, we can summarize it by saying the acetyl group from acetyl-CoA enters the cycle and leaves as CO2. So for all of these explanations, I've kind of been using my own words for them. So I'm really curious to see how you all have been summarizing these cycles. So in the comments below, let me know if you are using a different explanation and what your one sentence summary of each of the cycles is. So let's go ahead and zoom back out and fill out the rest of our columns. So when we have our acetyl-CoA enter and that acetyl group leaves as CO2, this is an exergonic reaction, and so delta G is less than 0. So overall, because it is losing hydrogens, it's losing electrons, this is an oxidation. So our acetyl group to CO2 is oxidized. While that is oxidized, we are also going to have a reduction of NAD plus to NADH, as well as a reduction of FAD to FADH2. And we're also going to make a molecule of ATP. And each of these reactions, you'll notice that I have drawn them as endergonic reactions. So the formation of ATP, the reduction of NADH, and the reduction of FAD to make FADH2 are all endergonic reactions, which means their delta G's are all greater than 0. So at the end of this stage, in terms of our energy management molecules, we have made 2 ATP, we have made 2 FADH2, and 6 NADH2 overall. And then if we wanted to continue our total column that we have for stages 1 through 3, at the end of stage 3, we have made 4 ATP, 2 FADH2, and 10 NADH. So again, this is a total for stages 1 through 3. Now at the end of the citric acid cycle, if we look at our carbon molecules, we have 4 CO2 that have left the cycle. So the very last question on this scaffold asks us, well, what happened to the carbons from glucose? And this goes back to that French fry question that I asked you at the beginning of aerobic respiration. So when we work out, when we break down food, the carbons that come from glucose, we exhale as CO2. And so we can say that those leave as carbon dioxide. So pretty amazing that is what occurs. So I'm going to actually stop here, and for the model extension that asks you to draw the electron transport chain in chemiosmosis, I'm going to go ahead and challenge you all to do that on your own. In addition, there is a flow chart with boxes and arrows that is another summary of the state of all 4 of the stages of aerobic respiration. So just another way to look at the information. I will post an answer key for the model extension questions on Moodle so that you can double check your work there. But as far as the modeling activity itself goes and the model application questions, and what happened in the Science Daily article, we're going to use the comment section of this video to discuss it as a class. So thanks for hanging out with me, everyone, and I will catch you in the next one, and I hope you have a great rest of your week. See you soon. Bye, everyone.